I start, I'd like to acknowledge some people right off the bat. My collaborators on this work that I'm about to present: Brendan Holland, Brian Bowen, Steve Carl. I did some of this work under the tutelage of Celia Smith, and especially like to thank the Hawaii Coral Reef Initiative for funding a lot of the research you're about to see. So there's 3 species of Opihi in Hawaii. They're all endemic to the Hawaiian Islands, and they're very important. They're important ecologically. There can be up to 50 per meter square on wave-exposed Hawaiian rocky shores. They can serve as aumakua, or sort of guardian angels for Hawaiian families, and they're delicious, and they cost about $15 to $30 a pound at the market. Because of these reasons, mostly because they taste so good, there's been a huge crash in the Opihi fishery in Hawaii over the past century. And what you'll notice is the biggest decline occurred in the early 1900s versus today. It's actually, there is a slight decline, but it's sort of leveled off at about 13,000 pounds of Opihi harvested per year. In the late 60s and early 70s, people started to realize that there was a problem with Opihi and how many there were in Hawaii. So a law was passed based on the research of Dr. Allison Kay, where they restricted the size that you could take. And the size limit is 1.25 inches, and this was determined to be an adequate size at which Opihi would be able to reproduce before they were harvested. And this is denoted by the dashed line on that graph. And what you can see is there's been no increase in the Opihi harvest, and I'll extrapolate that out to the Opihi populations in Hawaii, because the demand for Opihi is just as high or higher now than it's ever been. So we have to ask ourselves, what can we do now to help Opihi, because the size limits haven't worked. And I'm proposing today one answer is marine protected areas. Marine protected areas are often used as a tool for conservation, where you protect an entire reef or an entire area and you don't do anything to the organisms within that area. But you can also use it for fisheries management, in this case targeting Opihi, and specifically don't allow the harvesting of Opihi or anything that interacts with Opihi in certain areas. We would consider an MPA successful if not only we see an increase in Opihi and other organisms within the protected area, but the main idea is that we would see an increase in the recruitment of Opihi outside of the protected areas, because these protected areas are essentially protecting breeding adults. And the larvae of Opihi can disperse widely by swimming, and the hope is that they will restock the shores naturally that are being harvested. Hence, we can kind of throw out size limits and only work on protecting certain areas of Hawaii shores. So how does this work? This is sort of a little diagram of various pockets of Opihi along a shoreline or around Hawaii, and the pockets of Opihi circled in red we'll call marine protected areas. Those are protected, those are the breeders. The rest of the areas get harvested, and as you guys know, any area that's accessible that gets harvested for Opihi ends up with no Opihi by the end of the season. What we rely on is then the spawning of the Opihi in the protected area to repopulate the surrounding pockets of Opihi, and we're back to having a fresh batch of Opihi for next year's graduation party or other major luau event. However, it's not that simple, because we kind of have to know how far do the larvae go? Are they going to repopulate everywhere? And this is where population boundaries make a big difference. If we look at this little model and we say that there's a population boundary right in the middle, and we only have protected areas on one side of that boundary, what's going to happen is some of the pockets of Opihi aren't going to recover on a time scale that's relevant for fisheries management. So here's the big question: where do we put the MPAs in Hawaii? There's already some small MPAs within the main Hawaiian Islands, and the Northwest Hawaiian Island National Monument is one large protected area. Right off the bat, I can say that the Northwest Hawaiian Island National Monument, it's not going to help one of the species of Opihi because it doesn't occur there. So that's why we have 3 Opihi down the main islands, only 2 up in the northwest. And then the question is, for these other two Opihi, is the Northwest Hawaiian Island protected area going to help or not? And to do this, we need to know where are the Opihi going. And this is the dispersal phase of Opihi. You can see it's pretty small, less than a millimeter, about 1/10 to 2/10 of a millimeter long. And it's very difficult to tag those, and even more difficult to recover them after you've tagged them. So we kind of can't do that. So we use DNA. And here's how it works. I'm going to try to make this understandable to just about anybody. Think about hair color. There's a gene for hair color in people. You might have black, brown, red hair. We kind of do that with Opihi, but instead we look at a part of the electron transport chain, in this case called CO1, and we look at the blonde molecule and the brown molecule, and that's represented by the A, B, C, D, E, and F on the bottom of that graph. And on the y-axis or vertical axis of the graph is a number of individuals that we sample from each area that exhibit each one of those alleles or hair colors, if you will. Now, within a population area, we expect to find the same exact frequency distribution of these alleles because there's free interbreeding. So in every one of these pockets within one of the populations, you should find the same frequencies. When you go across a population barrier or barrier to dispersal, you'll find a different frequency distribution, and this is exactly what we do to determine where are the population breaks for any species. So based on the mitochondrial DNA CO1 gene, we found these breaks in the Hawaiian Islands. One between Gardner Pinnacles and Mopupapa, one between Kauai and the Northwest Hawaiian Islands, one between Kauai and Oahu, and then Molokai came out isolated in one of the species. So here's our first picture of kind of how are the populations of Opihi divided up within the main Hawaiian Islands and the Northwest Hawaiian Islands. And the first thing that we can kind of take away from this, see the pointer right here, these two species, the only two that occur in the Northwest Hawaiian Islands, there's a barrier between the Northwest Hawaiian Islands and the main Hawaiian Islands. So there's going to be